Rename.
Has it come?
Yes, sir.
Okay, good.
Hemant,
Yes, sir. Tell me.
yeah, Dr. Tyagi is there.
Namaskar.
Namaskar, Tyagi.
Namaskar, thank you, sir. Again, Rob.
Namaskar. So you want me to check the slides?
Yes, sir.
All right, so I will do it. Yeah, is it visible?
Yeah.
Great, great.
Okay, so I will keep it minimized mode.
You can check one or two slides, sir.
Okay, okay. Maybe it is loading. Just a minute.
Yeah.
Can you make this a little big, Dr. Sir?
यहाँ से तो big तो नहीं होगा और it is on the full screen.
Acha. Okay.
Yes, it is on the full screen, sir.
But then why I am seeing it half the screen?
Sir, because the panelist, all the panelists are there on the right side. Maybe if you put it down.
Okay.
Okay, that I will minimize if it starts moving. Okay, that's
Yeah.
the first issue.
Yes, yes.
I don't want to fiddle with it. Let it be there.
No, just wait for a minute. Otherwise, I will try in different way.
Yeah, because it is almost 47 slides, so it may take a bit of time to
Yeah, but not
to
that
upload.
much.
Yeah. Hello. Good afternoon.
Now it is moving.
Yeah. It's perfectly fine. And we can read this very nice.
Very fine. So that is one, and then you want me to minimize this, which I will do.
I'm not interested in that. Thank you.
So now everything is fine.
Yes, sir. Everything is fine. Perfect.
Okay.
Yeah, it looks
So
very nice.
okay, I think we are set.
Yes.
Hemant, can you put the name of Dr. Tyagi?
I was also to tell you that because panel name should be Akhilesh Tyagi.
Okay.
Good afternoon, sir.
Good afternoon, Mukul. Good afternoon.
Yes,
Little early. Should I join after 5 minutes?
yes, sir. You may
I'll
join
do-
after 5 minutes.
Is the video?
Good afternoon, Alok ji.
Good afternoon, Ken. Good
are
afternoon, Dr. Tyagi. Good
Good
afternoon.
afternoon.
अभी तो
Namaskar.
5-10 मिनट हैं.
हाँ 5 मिनट है.
So we'll I'll just join back.
We were just testing everything is okay.
Okay. Chaliye, I'll join back in 5 minutes.
Very
Okay.
fine.
Okay.
Professor Tyagi, you must, I would like to ask, utilize some time with you, sir.
Yes, yes.
I was wondering, you must be having in your department gel dog system.
Gel dog system. Yes.
Okay, we might be requiring some readings to be taken, some pictures to be taken.
Okay, we will see that.
Sure.
When you need it, then tell me. Okay.
Yes. Sure. Okay. Thank you, sir. Thank you.
डॉक्टर दास, आप
Yes.
जब बात करेंगे तो इको हो रहा है.
अच्छा.
हाँ, right now it was okay, but last when you spoke to
Huh.
it was echoing.
Okay. It's quite possible that all the microphones are on. That might be the reason that.
Correct. Possible. Do you have two microphones on by any chance?
No. I have only one, sir.
If you have two, it will create a problem.
Yes. Two microphones are there in the conference room, but they are kept at distance apart.
Sometimes it attracts. Sound can travel easily.
Mm-hmm. Dr. Tyagi, you can maybe minimize your slideshow.
Okay. I may have to go out of it to minimize it.
Okay. Okay. So रहने दीजिए फिर.
So let it remain as the title slide for the lecture.
So there must be some options, sir.
No, I don't see that in my computer at least. I may have to go back and enter into this slide. So that's the only thing.
If you try to, there is a button called escape.
Well, that is the point. That is what I am doing. If I escape, here it is.
Yeah,
Okay. So now
there is
it
a
goes in this mode, and then I can see here it is.
dash sign on the top right corner.
I have to open it.
That's going to minimize, no?
Yeah,
Yeah, minimize. It has already minimized, but
Correct. I
something.
think already minimized.
Yeah, but something else is coming. Screen is not coming. That's the point. Screen I have to enlarge. That's the point.
I think PhD people have joined with their logo.
view option.
I hope you can go back to your frame number 1.
Yes, it is visible.
Yeah,
No.
all the things.
Yeah, so this will be possible.
Okay.
Yeah, it will be possible.
Oh, then that's good. Yeah, you can go now.
Hmm. Uh, Good afternoon, everybody. Sir, can we start the program, sir?
Yes, please start.
Dr. Meenu,
Yes, sir.
please take care of all the proceeding.
Good afternoon. Respected Professor Akhilesh Tyagi, Meghnad Saha, Distinguished Professor of National Academy of Sciences, India, Shri Alok B. Sriramji, Vice Chairman, Governing Board, Shri K.N. Rao, Senior Member, Governing Board, Dr. Mukul Das, Director, Shriram Institute for Industrial Research, invited dignitaries, ladies and gentlemen. I, Dr. Meenu Talwar, scientist, Shriram Institute for Industrial Research, take the privilege to welcome you all to this 4th webinar in Dr. Bansidhar Science, Technology and Innovation Lecture Series. Dr. Bansidharji was closely associated with Shriram Institute for Industrial Research as Vice Chairman for over 3 decades. He was a great visionary and a famous industrialist of his time. Dr. Bansidharji believed in capacity and capability building in the country through indigenous R&D, thereby helping in the progress of the nation. He was a mentor and always provided ample opportunities to hone the skills of his staff and to continuously improve their knowledge. He not only nurtured the entire scientific community and their work, but he also taught us to remain humble by adopting a value system that enables us to become good human beings first. Today's lecture is being delivered by Professor Akhilesh Tyagi, known globally for his excellent work in the area of genomics and biotechnology. In today's lecture, Professor Akhilesh Tyagi will share his vast knowledge with us through his lecture entitled Diversifying Crop Genome. May I now request Sri Alok Sriramji for his welcome address.
Thank you, Binu. Can you hear me? Can you hear?
Yes,
Yeah.
Okay, thank
We
you.
can-
Respected Dr. Akhilesh Tyagi, members of the Board of Governors of Shri Ram Institute, Mr. K.N. Rao, Dr. Mukul Das, and ladies and gentlemen. A very warm welcome to our Dr. Bansidhar Science, Technology and Innovation Lecture, which is the fourth in its series. Shri Ram Institute has instituted this series of lectures in memory of its illustrious, long-time leading light, Dr. Bansidhar. Today, once again, we are coming together to listen to another thought-provoking oration. Shri Ram Institute was founded by Sir Shri Ram in 1947. This unique institution is independent, not-for-profit, self-sustaining, multidisciplinary, and committed to service of the scientific infrastructure of industry. It has always kept abreast with the technological advances and undercurrents through its existence of over 70 years. If it had not, it would not have survived. This adaptability is ingrained in its ethos from the founder himself and has kept shining by the leading lights that followed him as well. The main area of expertise in the institute are material sciences, in which polymers and composites, hydrogen research are prominent. Biological sciences, including toxicology, food sciences, microbiology, molecular biology, environmental sciences, civil engineering, radiation center for sterilization, etc. Today, our lecture is being delivered by Professor Akhilesh Kumar Tyagi, a renowned expert who has made enormous scientific contributions in the field of genetics and plant molecular biology. The detail of his accomplishments will be enumerated by Dr. Mukul Das, Director, Shri Ram Institute, when he will be formally introducing Professor Akhilesh Tyagi in a few moments. The title of Professor Akhilesh Tyagi's lecture today is Diversifying Crop Genome. Shri Ram Institute has had a long relationship with genes and genomic studies. As early as 2001, the founder memorial lecture of the institute was titled A Reconstruction of the Human Genome and Its Implications. It was delivered by renowned Dr. Eugene W. Myers, Director, the Max Planck Institute for Molecular Cell Biology and Genetic Systems Biology Center, Dresden, Germany. Through this lecture series, we remember a great personality, Dr. Bansidhar, who besides being a successful industrialist, contributed to building a strong science and technology foundation in our society, working from outside any government framework. He was the leading light that inspired Shri Ram Institute over a period of 3 decades during his time as Vice Chairman of the Board of Governors, besides being the motivation behind numerous other educational, technological, and industrial endeavors. Dr. Bansidhar, who passed away on June 26, 2004, is still remembered as a person with keen interest in scientific research, administrative acumen in organization building, and a man of compassionate heart that had a special place for young scientific talent. During his tenure, the Institute ventured into several new avenues. The Applied Radiation Center, Toxicology, Southern Regional Branch of Shri Ram Institute, Bangalore, the Environment Division, etc. were added during those times. Today, as we remember Dr. Bansidharji, it is worth recounting that he contributed a great deal in various areas of growth of the country, such as industry, education, industrial research. He was the president of FICCI, the Federation of Indian Chambers of Commerce and Industry, president of PhD Chamber of Commerce, president of Indian Sugar Mills Association, President, Indian Chemical Manufacturers Association, and also the Badminton Association of India. He was a member of the Center of the India International Center, a governing council member and treasurer of the Shri Ram College of Commerce, and chairman of Indraprastha College, Ramjas Foundation, and the Indian Association of Deaf and Dumb, amongst many other social and educational institutions. His simplicity and intense compassion was visible in his effort to help all. He was warm-hearted, caring, and always ready to lend his wise counsel and render help to those who approached him. He was a person with rare compassion, especially for scientists, whom he always placed above all others. Shri Ram Institute, which was founded by Lal Shri Ram and Dr. Bansidhar nurtured it, is still carrying on the vision which those illustrious personalities had passed on. It is our desire and passion that the society and nation should be impacted positively and be benefited from science and technology that is rooted in the Indian soil and founded on long Indian traditions that burn bright through the ages. Nothing can be achieved without thoughtful and deliberate planning and efforts, and to build a nation that progress on scientific innovativeness, we need to inculcate first a culture of innovation. To achieve our place in the 21st century, we need to build a strong science and technology foundation on which the thought process or the operating philosophies of our nation rests. We at Shri Ram Institute are fully committed to this ideal. Thank you.
Thank you, sir. I now request Dr. Mukul Das to introduce today's speaker.
Thank you. Thank you, Meenu. Respected Shri Alok Shri Ramji, Shri K.N. Raoji, Professor Akhilesh Kumar Tyagi ji, guests, delegates, colleagues, ladies and gentlemen. It's indeed a great pleasure for me to introduce today's speaker, Professor Akhilesh Kumar Tyagi. Although I personally feel that he needs no introduction to anyone who are in the field of plant molecular biology because of his illustrious contribution to plant genetics. Nevertheless, for the sake of the much diversified audience that we have today, I would like to recount on his major aspect of his personality and credentials. Born in 1956, Professor Akhilesh Kumar Tyagi received his PhD degree from University of Delhi in 1983. Professor Tyagi works in the Department of Plant Molecular Biology, University of Delhi, as Meghnad Saha Distinguished Professor of NASI. He has earlier served as Senior Professor at University of Delhi, Director of National Institute of Plant Genome Research, New Delhi, and Executive Director of National Agri Biotechnology, Food Biotechnology Institute at Mohali. Professor Tyagi is well known for his contributions towards crop improvement in India using molecular physiology, genomics, and transgenics. His research has widened scientific understanding of neo and sub-functionalization of regulatory gene families in plants during evolution. He has been among the group of scientists of the country who worked on genome-wide sequencing, particularly of rice, tomatoes, and chickpeas. He has worked on gene and regulatory elements of rice cultivation and investigated the transcriptome profile of crops and was successful in discovering a new stress-associated protein gene family in plants. His research has resulted in over 250 publications and he has mentored over 120 postdoctoral fellows, doctoral and research scholars. Professor Tyagi is a recipient of several awards and honors. Among the many, to name a few, are J.C. Bose National Fellowship Award, National Bioscience Award, Nasi Reliance Industries Platinum Jubilee Award, etc. He is a fellow of all the national science academies of the country and is a fellow of the Third World Academy of Sciences, and also had the distinction of serving as the president of National Academy of Sciences, India. This is the fourth lecture of the series in Dr. Bansidhar Science Technology Innovation Lectures. And we have listened to very profound talks from three different eminent personalities in the previous lectures. Today, Professor Tyagi is talking on diversifying crop genome, and we are looking forward to yet another equally profound and thought-provoking one. With this, I call upon Professor Tyagi to deliver his address or lecture. Thank you very much.
Thank you. Thank you very much, Dr. Das. Let me put my slides on.
Sure.
So they are visible on the full screen and I am audible.
So you are audible, your slides are not there, sir.
Slides are not there. Oh God. Are they there now? No?
No.
This is what I was worrying. Just a minute. Let me see. I think on the screen, can you minimize your screen? Because the same thing was functioning earlier.
Dr. Meenu?
Where is Dr. Meenu?
एक मिनट वो वीडियो ऑन करना पड़ेगा ना यहाँ से होगा इसमें जाओ.
Can I again go to share screen?
You have to go to share screen.
Yes, sir, you need to go to share screen.
Okay, again I am going. So here it is.
Yes, sir.
Yes.
So you see, we have shared in preparation and I have to share it again. Okay, let me make it full screen.
Right.
And now it is visible?
Very much.
Very fine. And it can move also. Very fine. So thank you, Dr. Mukul Das. Honorable Shri Alok Shriramji, Dr. K.N. Rao, ladies and gentlemen, distinguished luminaries, members of the governing board of the institution, it is my pleasure to give this fourth Dr. Bansidhar lecture. Dr. Bansidhar is known to be a transformational and visionary leader of industry, education, and industrial research. His contributions are there not only in the corporate sector, but all over the India and even cross the boundaries of our country. And therefore, I feel humbled to talk in the name of Shri Dr. Bansidharji. And for this purpose, I have selected a topic which is diversifying crop genomes, which connects the science to the masses of the people because it is a main area of research and investigation in agriculture. Now, all of us know that work of Rosalind Franklin, which resulted in this particular X-ray crystallograph, which is shown in the middle, was looked upon by James Watson and Francis Crick, and they could develop the model of DNA, which was at that time established as the genetic material, and that was in 1953, exactly 69 years earlier to the present time. And from that point onwards came into existence the central dogma of biology, in which we learn that DNA makes RNA with the help of DNA polymerase, which is an enzyme, and then this RNA is translated into the proteins with the help of ribosomes. So that has been the original and the fundamental form of the central dogma, which was thought by Nobel laureate Francis Crick. Of course, later on, we learned that RNA can sometimes make DNA also. So that was modification of the central dogma, but in most of the eukaryotic cells, the central dogma of biology stands true even today. At the same time, we learned that all organisms have DNA, which is the genetic material, and this is inherited from one generation to another generation, which we call as vertical gene transfer. So be it bacteria, archaea, or eukaryotes, we have vertical gene transfer from parents to progeny. But also it was known in recent years that there is transfer of DNA from one organism to another, and even in between the kingdoms which are there in the biological sciences. The classical example is the transfer of the plastids and mitochondria, which are considered to be origin of bacteria, coming from bacteria, which have been transferred into the eukaryotic cell, and they perform very important function in the eukaryotic cell, like photosynthesis and respiration. So mobility of the DNA vertically as well as horizontally has been established. At the same time, the people in the area of plant sciences have started to genetically modify food long back, knowingly about the genetics and sometimes unknowingly about the genetics. The very early efforts were meant for selective breeding by way of cross-pollination, in which the DNA of two organisms are mixed, and the resultant hybridization forms the new organism, which is a result of combination of the genomic component of these two organisms. This picture shows that how present-day maize has been developed by plant breeders from its progenitor, that is teosinte, by way of selective breeding, cross-pollination, and hybridization. This has been a well-established and long-known procedure, but in 1983, three scientists, that is Marc von Montagu from Belgium, Mary Del Chilton from USA, and Robert Fraley from USA, established a new method of transfer of DNA piece from one organism to another. And in that way came into existence what we know as GMOs, or genetically modified organisms in the agriculture area, that is the crops. And for that work, they were given in 2013 the World Food Prize. Of course, it came after 30 years, but has been an important recognition of this technology, which was developed by these three people. Now, by using this technology, a lot of GM food has been produced and has grown in last 20 years in the field. And this map of the world shows that overall, there are about 180 million hectares of the land being cultivated by GMOs in different countries. Of course, first in this race is USA, followed by several countries in Latin America, and in India, which stands at the 5th level, there is a single GMO crop which is being grown, and that is cotton. and this itself is being grown in about 12 million hectares. So you can consider that every year world is growing more land with transgenics than what the total arable land is available for Indian farmers. Now, in this particular case, in the last 20 years or so, there has been tremendous impact of this on business and industry, as well as economy of the world. Now, if we see that what transgenics have contributed, about 225 billion worth of the biotech crops have been contributed in last 20 years. Also, because of overproduction by these crops, about 213 million hectares of the land has been saved from cultivation in these 20 years. Also, there has been an impact on the environment because less amount of pesticide has been used. About 76 million kilograms of less pesticide has been used for growing these crops. Also, the CO2 emission has been reduced equivalent to about 5.3 million cars off the road per year. And most importantly, about 17 million farmers have benefited from them, and their families, including about 65 million people, have found a sustainability in the present day scenario. While there is a positive sign for this, the limitations are also there. First one is, of course, regulatory limitation, but another one is that this technology has remained restricted so far to only few crops like soybeans, maize, cotton, and canola, and other biotech crops are very limited in the area, only about 1.8 million hectares, although they are represented by sugar beet, potatoes, apples, squash, papaya, and brinjal. You will see that on the basis of that, we have not yet touched the food grains, which are the pillars of the world food security. And therefore, not only world security, but also Indian scenario is to be looked into. Now, in this, we have to remember Green Revolution, which within 30 years of its existence, tripled the food production. And the poverty and hunger percentage was halved, as well as farmers' income and employment was enhanced. India is now capable of producing more than 300 million tons of the food grain by itself and is food sufficient. But we also know that in next 20-30 years, we need to produce about 50% more of the food grain. And this food grain has to come from limited land. And also, we have to, along with this, perform or contribute to the sustainability goals, which have been defined by United Nations. And out of these, I would like to draw your attention to goal number 1, 2, and 3, which is no poverty, zero hunger, good health and well-being, also affordable energy, climate action, life below water, and life on the land. And these sustainable goals cannot be completed unless we have contribution coming from the agriculture sector. Now, this scenario has to be achieved in a situation where environmental degradation and social disruption are taking place. We know as of now that ecological degradation leads to loss of livelihood. And when livelihood is lost, then farmer families start to move to urban areas, and we have creation of the urban slums. Sometimes only the male folks move, and therefore there is feminization of the poverty. And this leads to social disruption, because of which we have urban slums with increased food insecurity and neglect of the farming activities. This vicious circle completes again and again, and therefore we have to take care not only of social disruption, but also of the environmental stresses, and produce from the same land area an amount of food grain which can sustain us on this planet, that is Earth. Now, in this case, excuse me, a new science has emerged, and this is called as the genomics. Now, what is genomics? Genomics is to compile genome sequence of organisms and establish location of all genes and regulatory elements, also establish function of all genes and regulatory elements which tell the genes where to express and what to do. And now within these genes, there are brothers and sisters of the genes which we know as alleles. And these alleles sometimes could be better and sometimes could be inferior. So we have to identify not only genes and regulatory elements, but also identify superior alleles. And then by identifying superior alleles, we have to deploy them for agriculture, health, and ecosystem. So that is the potential of the new technology, which we know as the genomics. Now, in this genomics, we start with the genome of the organism, be it plant or human genome, in which are there in the form of chromosomes in our cells. And then we prepare a genetic map. And on the genetic map, we align the cloned fragments of the DNA from the cell. And out of these cloned fragments, we select the minimum tiling path. Now, each of these clone in the minimum tiling path is sequenced and then assembled in the form of the sequence of the whole chromosome, which is anchored to the chromosome. So that is the approach which is called as map and sequence approach. On the other hand, we can also take out now the whole DNA, cut it into small pieces randomly, and by doing next generation sequencing, we generate huge amount of genome sequence data very rapidly. And with the help of computation power as well as artificial intelligence, put together the assembly of the genome. Of course, we should understand that in genome, there are some overlapping regions, and this leads to the incorporation on some gaps in the genome when such kind of assembly is prepared, and that need to be further confirmed. So there are two approaches. One is very rapid, but another is systematic. Of course, we worked first on this technology and later on adapted the other technology in the hybrid mode of the genome sequencing that we have done in India. Now, first genome which we sequenced, and this was the first high throughput genome sequencing done in India, was the genome of rice as part of an international consortium which had 10 nations involved in it. And this effort was supported by DBT and ICAR. And here I show you the letter which was given by Shri Atal Bihari Vajpayee ji at the time of release of the rice genome sequence in Japan, because Japan was the leading country of the consortium. And the work was appreciated by him greatly, including the work of the international community as well as Indian laboratories. Of course, this work was steered by Dr. Manju Sharma, who is the chairperson of the Board of Governors and has been nurturing this science for a very long time. And actually, she initiated us into taking up this particular project. And this is the delegation which was present at the time of the commemoration event in Japan, which was held at the time of the release of the rice sequence. Of course, we sequenced rice genome along with 10 nations, but we continued and followed on tomato genome sequencing, as well as more recently with international community on the wheat genome sequence. But then we asked the question, can India do the sequencing by itself? And therefore, the teams were assembled at NIPGR, where I worked as director, and the chickpea genome was sequenced by Indian team independently. And later on, of course, not only the Deshi chana, but also Kabuli chana and also the wild type chana were sequenced. Also, Indian scientists have contributed, or not only contributed, but sequenced by themselves genome of pigeon pea, finger millet, pearl millet, tulsi, neem, and mango. And as of now, worldwide, we have about 400 genome sequences of the plants, out of which most of them are in the draft stage. And the gold standard remains the sequence of rice in crop plants and Arabidopsis thaliana among all the crop plants which have been sequenced so far. Now, I would like to bring you to a stage which will happen in the coming few years. It has been planned that there should be a project called Earth Biogenome Project, and this will analyze the whole Earth biodiversity of the cellular organisms and would be helping the most crucial scientific and social challenges. Now, in this project, sequence of about 1.5 million species will be generated to revolutionize our understanding of biology and evolution, conserve, protect, and restore biodiversity, and create benefits for society and human welfare. I should also mention that this information is very much necessary for doing any genome editing of any organism, and genome editing is being considered one of the most powerful techniques to diversify the genomes. Now, coming back to rice, as soon as we sequenced rice genome, we found that 12 chromosome sequence which we have generated has got a lot of duplication. Actually, 60% of the genome was duplicated, and we established that this duplication happened about 60 million years ago when dinosaurs disappeared. And we consider that there was great change in the environment which initiated the process of genome modification, which led to the duplication of the genome which is represented in rice in the present day scenario. Also, we started to learn that how wild type rice differs from the cultivated rice. As you might know, the wild type rice is perennial, whereas present rice is annual. In the wild rice, we have undesirable characters like seed shattering, but now seeds are non-shattering type. Also, the habit and the growth characteristics of rice have changed over the period of time as present day rice, cultivated rice developed. You can also see the seed color. Here it is red and not that desirable, whereas the present rice is white and is a desirable trait. So questions came that how this has happened over the period of time, and by genome sequence available, we learned that there have been mutations in certain genes which led to this kind of improved trait availability in rice. For example, the shattering of rice, as you see here, in comparison to non-shattering of the cultivated rice, has been due to a single base change in DNA, in which we could see a change of G to T in a gene which codes for a transcription factor having homeodomain box in its architecture. Also, the trait of the seed color from this red pericarp to the white pericarp was result of a deletion of 14 bases in one of the gene which codes for another transcription factor known as the helix loop helix containing protein, and this led to this. So mutations have created desirable traits, and human beings have selected them and later on introduced in desirable varieties which are cultivated and eaten. So this is what we started to gain. But these were not few genes. We reported 30,000 genes in the rice, out of which 15,000 genes are such which are present in our body also. And another 15,000 were such which were plant specific or rice specific, and they make a mustard plant different from rice. So these kind of genes were identified. And then we moved to know which genes express where. So all 30,000 genes do not express or do not make transcript in all the cells, rather they express at a specific stage of development. Like what I am showing you is a set of genes which does not express in leaf, root, or seedling, or in panicle. or inflorescence or flowers, but it expresses in seeds and also does not express in stressed plants. So we could know that these must be the genes which are responsible for forming the seed. And in that way, we can define set of genes which are responsible for forming different organs or for developing the plants into a particular shape. Now, these plant genes expressed in a particular organ, like here I am showing you the panicle specific 3 genes which are expressing here, where colored boxes are there. But this is done with the help of a regulatory sequence. So we would like to know whether regulatory sequence is really able to confer this trait on any other gene. So we take the regulatory sequence and a reporter blue color producing gene. And now you can see that by raising a GMO, we can prove that yes, this regulatory sequence does not work in leaf or root, but it works in the flower. And in flower also, it works in the male organ, that is anther, and also in the germline, that is the male sperm and male pollen grains. So we could define the function of regulatory sequences which control expression of the gene at any particular stage. And by taking one such regulatory protein, regulatory element which expresses in the anthers, we raise transgenics by fusing it with an enzyme from bacteria, that is L-ornithinase gene. Now this enzyme it is able to convert a precursor of herbicide into a herbicide, which will kill the cell. So we put this gene together and introduced it into rice. And here you see that if we do not have the precursor of the herbicide sprayed on the plant, then pollen development is normal, female organ development is also normal in control, as well as transgenic plant which were expressing this gene. But if we spray this plant with the acetylphosphinothiazine, which is precursor of phosphinothiazine herbicide, then in control where gene is not expressing in anther, there is no problem. But in the transgenics, we see that the male organ is having the pollen grains which have died. And therefore, we could create male sterile lines by this kind of genetic engineering of a gene with the help of a regulatory sequence. And male sterility is a very important trait in the hybrid seed industry, because that is one of the most laborious tasks to do cross-pollination, and male sterility helps in the cross-pollination process with great ease, and therefore a trait of utility for economically production of the crop was developed. A way was found for that. Also, we found out some other interesting genes, like here is a helix loop helix gene, which itself creates sterility. So we don't have to depend on the genes from bacteria, but plant genes are also there. If their expression is affected or negated, then of course there will be sterility in the plants. We also looked at some genes which bind to a regulatory sequence but communicate with the machinery in the cell which makes the transcript. That is the first step of the central dogma. And we found that when we do that or kill this particular activity of the gene, then plants are dwarf. And not only they are dwarf, their panicle or flowers are also very small in size and create male sterility. Not only that, the seeds which are produced by this are also of a smaller size. And therefore, 3 traits: the size of the plant, the panicle or flower development, the size of the seed is being controlled by normal function of a single gene. If this activity is affected, then the 3 traits are affected together. How that happens, we found out that this gene, mediator 14. Mediator 14 interacts with different transcription factors, and they in different organ, either in leaf or in seed or in anther, ultimately affect the expression of the genes in such a way that we have the impact on development of these organs. So we learn about regulatory sequences, we learn about the genes which are responsible for formation of organ, we learn about the genes which are communicators and have larger impact on the expression of the genes. But it is not always that we are getting negative effect. We went further and analyzed a gene which is responsible for degrading undesirable proteins. And this particular E3 ubiquitin ligase gene, when we expressed in rice, then when we expressed in rice in such a way that the main activity of the gene is negated, then we could find about the clear-cut increase in the seed size. There was an increase in grain width up to 18%, 6% grain length, and overall grain weight increased from 13 to 42%. So positive trait could also be conferred, and we identified genes by this particular technology by introducing the genes inside the plants by way of genetic modification. Of course, another issue which we addressed was the stress condition. And this picture shows you that how production about 1 kilogram of rice requires about 3,000 liters of water. So that much water is consumed, but at the same time, for producing 1 kilogram of beef, one requires 15,000 liters of water. But what we would like to have is to produce rice with maybe 1,000 or 2,000 liters of water, or even less. And therefore, we looked for such genes, and we found one gene which is induced rapidly by cold, salt and water, mechanical wounding, as well as heavy metals and by submergence of the plants. We used this particular gene to overexpress in rice, and we could find that if we stress normal rice, do not give water or give very little water, then the number of the seed reduces by 90%. On the other hand, in the transgenic plants, this reduction is only about 20 to 30%. So we could get a gene which is able to provide 60 to 70% of the seed grain establishment in the soil moisture of 8 to 9% for 15 days. So drought tolerance trait could be conferred by such kind of the genes if they are discovered in rice. Of course, so far I have been telling you about the technology which is called as reverse genetics. And why it is called reverse genetics? Because we have the gene first, and then we connect it with the trait. But there is another genetics called as forward genetics, in which we start with the trait, like more roots, better panicles, good quality seeds, and then we try to find out which genes are controlling it. So this reverse forward genetics approach also we have made use of, and we identified by using genome-wide association analysis between the trait and that of the genes, 30,000 genes in rice. We identified about 23 new genes, out of which 13 were new and 10 were already known. So 23 genes were identified which are controlling the seed traits in rice. So that is the power of this technology. Of course, we also learned that there are some genes which are not present in all varieties. And this concept is known as pangenome analysis, and we identified by using available sequence of 3,000 rice genomes about 25% genes which are not present in all the accessions of rice. And by using these genes, we identified further 8 genes which control gene length, grain length, grain width, length to width ratio of grain, and 1000 grain weight. So new genes can be identified sometimes from new varieties and may not be present in all the varieties. So we have to depend on the natural genetic variability as it is available to us in the germplasm available in gene banks. Of course, Indian scientists as well as world scientists have made use of genomic information to improve the rice, and they have been able to pyramid the genes for resistance for disease. For example, here there are 2 genes for bacterial blight resistance which have been put together in the cultivated varieties of the rice in India. Also, there is effort to introduce bacterial blight as well as blast, that is fungal disease, as well as salinity genes together in the same varieties, and also to bring flood, drought, and salt tolerance together. So these kind of genes have been identified, and then they have been put together in the variety. And I understand that today about 3 million hectares of the land is growing such kind of rice in India also, because this does not require the regulatory processes, and this is similar to the precision breeding which farmers and the breeders have been trying for a long period. Only thing is that we are making use of the information coming out of the genome sequencing and then diversifying the genomes together. There is an effort in India to identify genomic sequences of more than 10,000 accessions, and then look for traits and genes combination which relate for biotic traits like bacterial disease, blast disease, false smut, sheath blight, and abiotic stresses like drought, salinity, submergence, and nutrition deficiency, as well as other grain quality traits. We, as I told you, worked on chickpea also, and in chickpea, by combining the technology which are available for the forward genetics, we identified first a gene which, or a brother of a gene, which provides the positive trait on seed size and nutrition. And then by using the parents, one having a small seed size and high seed number, another having large seed size and low seed number, we were able to move the desirable gene allele and improve the chickpea in such a way that it gave 20% more yield, 13% more productivity, and 15% better protein content. So that is the power of genomic technologies which are available, which include the genome-wide association, QTL mapping, molecular haplotyping, fine mapping, and of course the classical introgression of the genes by way of breeding. Now I would like to leave you with the final thought by introducing you the technology which we are learning commonly as the genome editing technology. This technology is most powerful in modifying the genes or any particular gene on the regulatory sequence, and is based on what we know as CRISPR and Cas9, a combination of a guiding RNA and a nuclease which will help the modification of the gene. And this technology has been developed by a lot of laboratories, but the major contributors were Emmanuel Carpentier and Jennifer Doudna, who got Nobel Prize in Chemistry in 2020, and you see them enjoying their Nobel Prize. But what I want to show you is to give 2 examples of the power of this technology. In case of wheat, there is the disease, fungal disease, which is providing, or which is there because of 6 genes. There are 6 genes in wheat, which are susceptibility genes. And by using gene editing, these were nullified. So 1 gene was nullified, 2 genes were nullified, 3 genes were nullified, 5 genes were nullified, but plant was still susceptible. And then the 6th gene was also nullified, and now plant became resistant. So it is possible that within a single plant, you are able to modify multi-genes together and confer such kind of difficult traits as I have told you for fungal resistance. One of the product which is already in the market in Japan is tomato. This is nutritionally important because it produces what we know as glutamic acid decarboxylase enzyme created GABA molecule. Now, this GABA molecule is considered to be nutritious as it improves health and also provides nourishment to our brain. So there are a lot of such projects, such products coming out. The issue again would be that whether we will allow deregulation of this technology and let farming community and industry also prosper, or we would like to put these conditions as we have put with GMOs earlier in place. The draft regulation is already out by Department of Biotechnology, but the final rules are yet to be established in India. We are waiting to see that at least some of the modifications by genome editing technology will not require such a cumbersome regulatory process as is required by GMO technology and is limiting that use of that technology. And therefore, till we are able to make use of GMO technology for diversifying the genome, we have other powerful technologies like the precision breeding, which is based on genome information, and also genome editing, which is also based on the genome information and the function of the genes. The last slide is to show that while we have done, or I have shown you this work representing some example from my laboratory, also from other laboratories in India and worldwide, what remains is a single fact that out of 30,000 genes, we know only about 5,000 gene function even today. So there is a lot of work ahead for identifying useful genes which are responsible for form, function, and sustainability of the crops. And not only we have to identify their function, we have to look for allele variability, find out brothers and sisters which are associated with superior traits, and then we have to combine or modify these genes and superior alleles to diversify and improve crops. So with this, friends, I hope I have given you the glimpse of the genomics technology and the ways to modify the crop genomes for improvement of the crop production and also to help sustainability of our food sector. Thank you very much for giving me this opportunity again, and I remain thankful to the organizers and pay my respect to Shri Bansidhar. Thank you very much.
Thank you, sir. Thank you so much for the thought-provoking lecture and highlighting the potential of genome or gene editing in crop diversification. Since there is no time for question and answer, I would request Shri K.N. Rao to propose vote of thanks.
Can you hear me?
Yes, please.
Respected Professor Akhilesh Tyagi, Shri Alok Shriramji, Dr. Mukul Das ji, leaders, ladies and gentlemen. It is a great pleasure to propose vote of thanks to our eminent speaker for today's very enlightening lecture. Professor Tyagi, indeed, your talk, Diversifying Crop Genome, was very interesting and covered very new areas like gene transfer concepts, contribution of biotech crops to food security, sustainability, and climate change solutions, and genome sequencing techniques. I can vouch that this talk was an inspiration to all those who are from the scientific community. At the same time, an informative one for those who are from other walks of life. This being the fourth lecture of the series, in Dr. Bansidhar Science and Technology and Innovation lectures. And like the 3 previous lectures in the series, is very engrossing. We had the privilege of participating and commemorating Dr. Bansidhar, who built Shri Ram Institute to its present stature. Professor Tyagi, we are grateful to you, and on behalf of Shri Ram Institute and all the participants here, please accept our heartfelt thanks. I thank Dr. Manju Sharma, our chairperson, Board of Governors of Shri Ram Institute, who has been leading with her vision and has been the inspiration behind today's lecture. Madam, thank you. Shri Alok Shriramji, with his great vision and passion for science and technology, has been the driving force behind these lecture series. He is the motivational strength for the young scientists, and the scholars of the Shri Ram Institute. We are grateful and thankful to you, and we will continue to look up to you for your inspiring leadership, sir. I thank Dr. Mukul Das, Director, and the Shri Ram Institute team under his leadership, whose tireless efforts made today's lecture possible in such a smooth manner. I thank Dr. Jitendra Singh, Director, and Mrs. Ladli Khatum and their team from PhD Chamber, who made this platform for organizing the event. A big thanks to each and every one of all the participants for taking out your valuable time and being a part of our program today. Thank you all.
Thank you. Thank you very much.
With this, we have come to the end of today's program. We hope that you will all join us again in the next lecture in this Dr. Bansidhar Science, Technology and Innovation series. Good evening to all and stay safe. Leave karna hai hame?
Yeah.
So we can leave now.
Yes, sir. Thank you very much, sir.
Thank you.